pleasure to introduce today's speaker, Dr. Ruth Ley, who comes to us from Tübingen, where she's the director of a microbiome institute at the Max Planck Institute there, I guess, Department of Microbiome Science. But I can't claim credit for bringing Ruth here. This really goes to my student Taichi. So Taichi is a PhD student who's interested in microbiomes and is hoping to finish this year, and so he's starting to look around for postdocs. And he asked me, well, how should I go about this? And I said, it's easy. You identify you identify first the field that you want to go into, and then you identify the very best field anywhere in the world who are working in this area, and you write to them. So Taichi did his homework and identified some of the top people in the field, and came back to me and said, well, Ruth Ley is just fantastic. And I saw that she's giving a talk at UC San Francisco on October 4th. and so I'm going to go to her talk and try to meet her. And I said, well, if she's giving a talk at UC San Francisco, let's bring her to the MVZ. So I come to find out that she's spending a week on the West Coast. This is the West Coast tour. And she gave a talk yesterday at UCSF and is giving a talk at Stanford on Friday, I guess. But miraculously, today, Wednesday, was open in her schedule, and equally miraculously was one of the few open dates we had in our schedule. So I wrote to her and I said, we'd be honored if you could come and give a talk here. Not expecting that it was going to be possible given her schedule, but it turned out that she was free. So we're delighted that she was able to come and be with us today. It was only after that I learned that she's a former MVZ student. So I had no idea. I know her as one of the leaders in microbiome research, but I have some quotes that I want to read from her about her time here. She said, I learned to catch lizards from Harry Green and the many talented grad student TAs at UC Berkeley. I took Natural History of the Vertebrates, IB 104, which is the best class ever. And then I took herpetology.
That's all right. I love them both.
This ability came in handy as an undergrad as I worked for MVZ grad student Chris Schneider, catching anoles on the island of Guadeloupe one winter break, and I spent a summer observing nesting behavior of a lizard whose name I forget, in the Chiricahuas in southeast Arizona. So apologies to the herpetologists in the audience. Although I took a wrong turn professionally after that, I still whip out my lizard catching skills occasionally. So the wrong turn that she took professionally led her to such fundamental discoveries as the role of the gut microbiome in things like obesity in humans, and the genetic determinants of the gut microbiome in humans. So this is absolutely fundamental research. To put it in perspective, she's published 10 papers that have been cited, each cited 1,000 times or more. So remarkable work. After leaving here, she did her PhD at Colorado, and then a postdoc at Colorado, and then a subsequent postdoc with Jeff Gordon at Washington University. From there, she was hired as a research professor at WashU, and then went to Cornell, where she was promoted to associate professor before moving to the Max Planck Institute as a director just this summer, I guess.
Yeah.
So Taichi did the right thing in identifying one of the leaders in microbiome research, and we're delighted to have her. Please join me in welcoming her to the MVZ.
So this would make a really great lizard catching stick. Well, yeah, it's really delightful to be back. I mean, I can't say enough about how wonderful it was to be at Berkeley. It's great to have Mary Power in the audience and see some of my professors from back when I was an undergrad. And yeah, I mean, you have such an exciting place, and I think you have to realize what an impact that has on the undergraduates who also, it was such a wonderful place to be during I'm getting all emotional. Anyway, it's really nice to be back. It's at Berkeley that the concept of diversity, just as in and of itself, became so interesting. And why is there so much of it, and what promotes it, and how does it keep going, and so on. And I think that my work with the microbiome is fundamentally about looking at diversity, mapping diversity, trying to understand what maintains diversity. And I happen to be looking at it mostly in humans at the moment, but I've also looked across some mammals in the past, managed to do that at a medical school at WashU, that snuck off to the zoo. And I know that a lot of you are doing that to a much greater degree, and so it's wonderful to see microbiome moving into intersecting with zoology as well and evolutionary biology. But for me, if I'm at all interested in this kind of stuff, or what drives my work really came from being here with you people. So when we think about what gut bacteria do for you, these are the big ones. They help you digest your food, ferment fiber, produce essential vitamins, break down toxic compounds, protect against invasions of things you might not want. There's evidence that they're very important for priming the immune system in early life, and that has consequences for lifelong health. And right now, at least, the understanding is that your mammals are born germ-free. So that might become a bit more controversial. But generally speaking, when a mammal is born, it's really devoid of appreciable numbers of microbes, and it's colonized very rapidly. This is a time series over 2.5 years of a particular individual, a baby, and you can see changes in abundances of different kinds of microbes over time, and the biomass goes up, and there's lots of important interactions in these early months that occur with the immune system and so on. And so you might, but this is environmentally acquired, and each individual has different exposures starting at birth from their family, from their environment, from where they live, from where they grow up, and so on. And so it's fascinating that this is left to something that this important organ comes from external sources, given how much it does and given how important it is to almost biology. And so we've been asking, why would you leave such an important process entirely to chance? And of course, there's a lot of organisms for whom it's not left to chance, and there's vertical transmission. But with mammals, that occurs to some degree because we colonize one another and we colonize our mothers and so on. But we've been asking just how much genes influence this process. So we know that the geneticists have been telling us that genes are important for phenotype. And we know as a field that, from transplanting microbes into germ-free mice and so on, that microbes are also important for phenotype, and different sets of microbes can influence metabolic phenotypes and different phenotypes. And so we asked this question of just how much do genes influence the microbes, and can that feed back on genotype? And to get at this question, we're looking at natural genetic variation that occurs in human populations. And one of the studies I'll tell you about now is one we did using twins. And so twins are quite fascinating because they're human clones, obviously. So these would be MZ twins that are clones. And a variation in any of their traits are due to environmental effects, whereas MZ twins only share half on average of their genes. And so differences here are going to be more environmentally driven, and when you could, for any quantitative trait that you can measure across twins, you can then model how much of the variation in a trait is due to genes or environment. And so, for instance, for any given twin pair, if you look at quantitative traits for one twin versus another, if you get a better fit for MZs versus DZ twins, that's the basis of working out heritability, and so you know that there's a genetic influence here. And so I'm sure that all of you are better versed in genetics than I am, but twin researchers have been doing this for years. And what you can get is an idea of heritability, so how much of the variation in a quantitative trait is due to genetic variation, and classic things like height. You can sort of work that out. And our question was, what about the microbiome? How much of the microbiome would be heritable, and then once we can identify heritable elements of the microbiome, can we then start to find genes responsible for driving those heritabilities? So I should say right away that there's been utter confusion in my field of microbiome studies because this word is so similar to the word inherited. And we're not talking about parents passing microbes to offspring. We're measuring the abundances of microbes across the twin population and looking at variance in the abundance. So we don't look at anything that's not shared in at least half of the twins that we look at. And we're looking at, are you high or low on Bacteroides theta iota omicron, or that's what we're doing. So we don't actually care if they were inherited. You could have picked them up from your dog yesterday. As long as they're there, we can measure them. That's all we care about is the variation in the relative abundance. That's the trait we're modeling. So twin researchers have been doing this for lots and lots of traits over the years, and these are the kinds of numbers you can expect to see. So height, 80% heritability in the twins population that we've been looking at, and other things are less heritable. And so our question was, where's the gut microbiome on this? Is it high, is it low, and what components and so on. So we worked with Tim Spector at King's College London, and he runs a large twin registry. If you're a twin in the UK, he will find you. He will enroll you. And so here are a bunch of their twins. And so they've been doing this for at least 15 years, and they've got something like, at least 15,000 twins enrolled, and many of them are genotyped, and they have lots and lots and lots of phenotypic information, and they work with a lot of people, so they're a great resource of phenotype and genotype information. And their twins come in once a year to the clinic, so they can be assessed for various metrics and their health and so on. And we enrolled as many as we could into this study where they donated fecal samples. So what I'm going to tell you about is results from looking at over 1,000 twin pairs. And so we've got 486 DZ twin pairs, over 600 MZ twin pairs. Many of them came in and gave repeated samples. They tend to be women, and they tend to be middle-aged, sort of 60 years old. I have no idea why. That's the type of people who are interested in participating in the study. And we have BMI information here. And the data we generated for over 3,000 fecal samples is 16S ribosomal RNA gene sequencing using the MiSeq platform. And so this gives us a phylogenetic readout of what's in those samples. And so we get about 80,000 sequences per sample that we then model using this ACE model of heritability, comparing all of these twin pairs to one another. So we take the 16S sequences, and then I think you're probably familiar with this, we bin them by type, and then you can say we've got so many of this species, so many of that species, etc., based on their 16S genes and how many sequence counts we got per person for those, etc. So to do this, we basically call up the twins, get them to give a sample, it's shipped to the mail, and then there's a lot of painful aliquoting of samples and buying another minus 80 every few months, which is really great for the chair of the department to have to deal with, and robotics and data analysis finally, and then this is what you get out the other end. So this is a phylogeny of the bacteria with some archaea. This is what our primers pick up. And what my student Julia Goodrich, is she on here? Picture? No. Oh, she was. I showed you a picture. All right. So this is work of my student Julia Goodrich, and what she did was for every single node in this tree, she's running that ACE model of heritability, and then she's coloring the branch in the tree according to the strength of the heritability. So this is the sort of typical types of bacteria you find in the Western human gut. A lot of firmicutes, some Bacteroidetes, which is a separate phylum here. We have some archaea, which are methanogens, and you can see that there's a lot of cool colors, meaning not heritable, but then we have specific branches popping out as heritable. So in this haystack, we've got needles, if you will, of specific groups of taxa that are giving us a heritability signal across these twins. Generally speaking, the Bacteroidetes don't, but the specific families in the firmicutes, and then the methanogens as well that we're picking up. And it's about 10% of the taxa that we see give us some idea of heritability. And so for those of you like thinking about statistics and sample sizes and so on, you can't really read this, but these are our taxa ranked by heritability. So that these are the heritability estimates of these dots, and they're coming down from higher to lower as they're ranked here. And what these lines are the size of the confidence intervals when we looked at 1,000 versus 3,000 samples. And what this is telling us is that the actual heritability estimates stayed about the same when we doubled the sample size, but the confidence intervals shrank. So this is it's nice to know that you could sort of redo a whole analysis and get the same thing again, and just improve your confidence around these estimates. And so I'm going to focus on the taxa that are at the top of the heritability list now. And tell you what we found out about them. So the sort of most heritable ones up here. First of all, so 3,000 samples sounded big to us, but it's not really. It's not very big for a GWAS study, and it's not very big for a sort of human genetics study. And so when you get these taxa as heritable, you sort of wonder, is that spurious? Is it not? And one of the ways we can look for validation is by looking at other published studies that have done sort of the same kind of things. And so there's a few published human studies now that are smaller, so in the Hutterites, for instance, or the HMP. And then there's some mouse QTL studies that have done the same kind of approach, but looking for genetic loci. And this is a table that you can't read, but what we did was go through all these studies and look for the same taxa and ask, do we see them in 2 or more studies at least? And the nice thing there is there are certain taxa that are showing up again and again across mouse studies and across human studies. So for instance, things like Heritibacter and Bifidobacteria. And so I think this is telling us that there's some associations that are even happening, that we see again and again, even across species and between mice and humans. And it'll be interesting to see once we start bringing in more animal hosts, just being able to see which ones track with genetics and hosts and which don't, and so on. And so again, there's so much natural history that needs to be done with understanding which microbes go with which animals and why, and why they associate with hosts and so on. But so we see a lot of these taxa replicating across some of these studies, which gives us some confidence. And so what are they? The most heritable is this thing called Christensen-Liaceae. And I really like bacterial taxonomy, but I've seen a lot of names, but this is one that I had actually never seen. And the reason it is because it had only been christened Christensen-Liaceae about 3 months before we found it. And so it was impossible to go back through the data, through published papers, and find out anything about it, because although the sequences have been detected, they've never been named, which is an interesting problem we have in my field, which is we only tend to talk about things with names. But the reason it had been given a name is because finally for that branch in the tree there was an isolate that came out of a lab in Japan. So I'll come back to this one. Charisibacter is another one that's not known much. There's not much known about it, but it looks like it's in the small intestine, perhaps. It's showed up in 2 different mouse studies, QTL studies. So that's one to perhaps look at for interesting biology. Bifidobacteria, which I'll come back in a minute, and methanogens. And I'll show you that methanogens and these guys are working together, we think, metabolically in the gut. So one of the analyses we can do with our repeat samples is ask about stability over time and on versus heritability. And so on this axis we have a measure of stability over time. Things on this end are more stable, and it's colored by strength of heritability. And so we see that some of our top heritable microbes tend to be temporally stable. So there's a huge range in stability, but you don't see the most heritable ones being unstable, and that's something sort of reassuring about that. So we can see which ones co-occur, and to do this we can look at counts of taxa across samples. and look to see which taxa co-occur across samples, and then build another matrix from that and display that matrix as a network. And I'm showing you this here now for our data set. We can see taxa co-occurring in the two major groups, and I'll focus on this one because this one includes a lot of our heritable taxa. So here the nodes are taxa, and they're clustered together if they co-occur across the samples. And they're colored by the strength of the heritability. So here's Chrysanthidiaceae, our most heritable family, and it's co-occurring with these other heritable taxa, including methanogens and a lot of other things that we know only from the fact that they tend to exist in things like fermenting bioreactors and so on. None of these have cultured representatives, only this group here. And we know that this the one isolate we have from this family makes a lot of hydrogen. We know these take up hydrogen and make methane. And so we're hypothesizing that some of these other ones that are not known in culture are probably also syntrose using hydrogen produced by this one. But that's a working hypothesis. What we noticed was that this co-occurrence network, this consortium, is enriched in lean people. So we can look at lean versus obese across our population. And now the nodes are colored by the strength of their association with leanness. So there's more of these things in lean people. We can go back and look in published data sets and ask where has Chrysanthidiaceae been found before. And we see it's typically associated with health. So in Pete Turnbow's paper of obese and lean twins, it was more abundant in lean. It's generally associated with health versus non-health. So here's some data on Crohn's disease and IBD and there's higher levels in healthy people. And this study came out just last year of close to 900 Dutch people, and Chrysanthidiaceae is associated with a low BMI and healthy levels of triglycerides, as are the archaea that co-occur with it. So we're seeing this come up more and more now, that this seems to be a healthy lean associated microbe. And so we thought we would test whether it could, if we could recapitulate that phenotype. So we know from Jeff Gordon's work that you can take a microbiome from an obese donor and put that into a germ-free mouse recipient. So these are raised aseptically, and you can gavage them with a fecal sample. It's like a fecal transplant. And if you do that in comparison with a lean donor, you'll find that you can transfer some of the phenotypes. So you'll end up with a fatter mouse, basically. And you can do this with lean and obese mice that are genetically obese or diet-induced obese. And you could also do this with lean and obese human donors to get the same thing. And so there's a lot of work into understanding how the microbes confer this kind of phenotype. But we were interested in seeing whether we could basically add Chrysanthidiaceae to an obese donor microbiome and end up with a leaner mouse than if we didn't add it. And this is the work of, Jill Waters here. So this is a single human donor with a high BMI, and what we do is we take the fecal sample and then we add in Chrysanthidiaceae or not. This is the type strain of the family now, the isolate. So we're adding the species to the mix and then transplanting the mice. And what we see after 3 weeks is that the mice that receive Chrysanthidiaceae they weigh less and they have less body fat as measured by DEXA. And so we've done this with heat kill controls. It's reproducible. We can see it in different strains of mice. And this is where we're at in trying to understand now why this is happening. And I can't actually tell you. We can discuss this later. Of the things we've looked for, these animals are not sick. I can tell you that. They're perfectly healthy. They have perfectly nice gut integrity and so on. But we're focusing on lipid cycling and also the metabolism, looking at metabolic products of the whole community that has this Chrysanthidiaceae and its consortium to try to understand how that's affecting the metabolism of the mouse when that's present. So this is a case where something about the genetic makeup of the individual is promoting a group of microbes, Chrysanthidiaceae and its syntrophic partners, and we think this is affecting phenotype in a healthy way here with lower BMI. So another thing we can do with these twins, because they're genotyped, is look for alleles that associate with specific abundances of bacteria in the gut. And so we've done this. We've run a genome-wide association study or analysis, and this is too small to get study-wide significance controlling for all tests that we do. So we're doing, we have 2 million SNPs, we have 600 traits, and we don't get study-wide significance for anything. We do get genome-wide significance for Bifidobacterium, which I'll come back to in a second. So these are some of our top heritable microbes. Christens Liaceae, we're not getting anything in particular. Nothing reaches genome-wide significance, so there's no sort of single gene here that's associating with this. But we are getting a signal from Bifidobacteria. And if we zoom in, it's a region on chromosome 2. And if we look at this particular region, we're picking up the region of the lactase gene, LCT. So SNPs at this position in the lactase gene will dictate whether or not you're lactase persistent or not, and that should predict whether you're lactose intolerant or not. And so, as you know, this is something that lactase persistence evolved a couple of times in human populations. It's one of the strongest signals of recent human evolution. And we see this also in the human microbiome data. So the Human Microbiome Project generated metagenomes from skin. And when they did so, about half their sequence reads were human. And we were able to take those and assemble them and call SNPs on them and run the same kind of analysis on 91 individuals, which is extremely small. But you can see an association coming out of that for the gut microbiota and this same position, the same area on chromosome 2 that includes the lactase gene. So in our data, we can see with the direction is that people predicted to be lactose intolerant or that with lactase non-persistence have higher levels of the bifidobacteria in the twins. And we see the same direction in work that was done by Emily Davenport and Carol Ober from Chicago. They've looked at the Hutterites, which are a founder population that's been studied for microbiome at 2 different seasons. So these people eat together communally, and you sort of that's nice for microbiome studies because you can constrain diet to some degree. But regardless of winter or summer diets, we see the same direction where bifidobacteria are increased in people with this GG genotype relative to others. So this is what we think is going on, but we don't know for sure. This is just a hypothesis, because what's happening is if you can keep making lactase, then you have low levels of bifidobacteria, but if you can't make lactase, then you have higher levels. And we think that these Westerners, like these English people with their tea, are putting milk in their tea perhaps, and the lactose is being utilized by the bifidobacteria, but no idea how true that is. That's the only sort of way we could think why you might have more bifidobacteria, which are known lactose utilizers in hosts that do not make the lactase gene. So this lactase persistence arose during the Neolithic period, when people shifted from hunting the kinds of things that you still have in your collection here to actually drinking milk and growing crops that were rich in starch. And so there was a big diet shift during this period for certain populations, including populations in Europe and Asia. And another one that's thought to have occurred during this time, a big sort of shift in human population genetic variation, was duplication at the AME1 locus, which is the gene that encodes for amylase. Amylase begins the starch breakdown process in the mouth. And Perry et al. A few years ago suggested that, based on looking at copy number variation across populations, that the mean shifted higher for people with a background of an agrarian background compared to a hunter-gatherer background. And this is what the data look like. Looking at hunter-gatherers versus people with an agricultural background, you see variation in copy number for this gene, but the means shifted higher for people with traditionally high starch in their diets. And this is thought to have been a result of selection from this big cultural shift to eating more starch. And we asked, looking at this, what would be the consequence for the microbiome to be at this end or this end of the spectrum? And so this is the work of Angela Poole in my lab, who enticed over 100 people at Cornell University to come in and have their AME1 copy number measured. And so this is the same kind of variation that we see on our campus, from high to from low to quite high, and then she enrolled a subset of these into our study. And so we're mostly comparing people at the low end from people at the high end of this distribution and asking, how do the microbiomes differ, or do they differ? And so we were worried that people who had different copy number might eat different diets, because it's supposed to, the amount of amylase in your mouth is directly related to this gene copy number. So the higher the copy number, the more amylase you make. And it's supposed to affect how you sense food, and bread's supposed to be more sort of central for you if you're at the high copy end, and you might prefer that. So we put everyone on a standardized diet working through the nutrition unit at Cornell. And we sampled them over the course of a month. So this is a 4-week study with a standardized diet in the middle. We took saliva and stool samples, had everybody write down what they ate and if they cheated while they were on this diet. We made sure that we knew what they were eating. And then we had paid them a lot of money in $100 bills. And they liked that a lot. And we're very compliant. So they came, can you imagine undergraduates being paid to eat? So this was the kind of menu they had. We made sure basically that they had starch at every meal, and they were allowed to eat as much or as little as they liked. And so we know that during this diet period, which is in the middle here, the starch intake was not different between the high and low group. And in fact, being on this diet didn't really change. There was variability from day to day, but it did make a big difference to how these people were eating. What we know from looking at salivary activity in the mouth was that the high group, as predicted, had higher overall salivary amylase activity, which is what you'd expect. So this is in saliva. And then if we look at their microbiomes in saliva, and this is a PCOA plot looking at overall diversity, we see maybe a slight shift, but for sure higher alpha diversity in the people with the high copy number, so higher species richness, if you will. And then we can ask which specific taxa differentiate high and low groups over time. So here are the taxa that we found differentiate high and low groups, and where it's gold colored, this means that this particular taxon was more highly abundant in the high group relative to the low group, and where this purple is the other way around, enriched in low. So generally we see taxa enriched in high, and you can see that they sort of come and go out of statistical significance. So here they're enriched, and here it doesn't reach significance, and here it does, and here it doesn't, and here it does, and so on. And so we see that it sort of comes in and out over time, which argues that it's important maybe to, when you're looking at the microbiome and you're looking for subtle effects, to look over time. You can see things come and go in and out of statistical significance, but it's always in the same direction. And this block in the middle where we had them on the same diet didn't impact these results. So in fact, we needn't have bothered to put them on the diet, is what we found. These two groups, so we're looking at the oral microbiota now, and we see Porphyromonas enriched. And Porphyromonas is known to be associated with bad oral health. And there's been some wonderful work going back and looking at plaque on people's teeth through the years. And you can see Porphyromonas coming into plaque between the Mesolithic and the Neolithic, and it's the same ones that we see same relatives of the ones that we see enriched today in the people with this high amylase copy number. So we think, although we don't know the oral health history of the people in our study, we think that they're at higher risk for periodontitis because of this. Now if we look at the fecal samples, there you don't see an effect of copy number on the activity of amylase in stool. So by the time you get to the hindgut, that's gone away. And in fact, it's sort of known that amylase activity in the large intestine is due to pancreatic amylase, which comes in from obviously from the pancreas. And the salivary amylase doesn't add to that significantly. So we're not picking up overall higher levels of amylase activity in the feces of the people of the high group. And we don't see a shift in beta diversity, and we don't see any differences in alpha diversity. But we do see differences when we look for specific taxa that differentiate the groups. Again, we see quite a few that are enriched in the high, which are along these lines here. And you probably can't read it, but many of these are actually from the Ruminococcaceae. And the Ruminococcaceae are known to break down resistant starch. We did some metagenomic analysis to look at functional genes, and we see paraplasmic alpha amylase gene counts higher in the high group. And these actually map back to Ruminococcus genomes. And so what we think might be happening here is if you have higher salivary amylase activity that's breaking down the simple starch, and those breakdown products are taken up by the host in the small intestine, that those are no longer available, or not as available as they in the high copy number hosts compared to the low copy number hosts. And we see an enrichment of bacteria that then are more focused on resistant starches, which by definition are not accessible by host enzymes. We wanted to see if there was a phenotype effect that we could pick up that might differentiate these two groups. And so we did germ-free mouse transfers of every human donor, then got its own mouse. And we're comparing now phenotypes of mice that received the microbiota from these individual people. And we did this for 4 different time points in the study. And for 2 of the time points, we see that the high copy number donors give us mice that have higher body adiposity. And for 2 of the time points, we don't see that. And these time points were when the people were off the diet, and these time points were when people were on the diet. And so this could be a coincidence, or it could be telling us something about the subtleties to the diet and what these people were eating that we couldn't access with the information that we had. So we see a genotype by diet interaction here and its effect on mouse phenotype, but again, we don't know what's driving this. And we've measured a lot of things, and we don't know exactly what's going on here. So this is another area where we need to dig deeper and try to see if we can understand mechanistically why these microbiomes are giving us different phenotypes, and especially when they're coming from the donors at different times. So we know from this that copy number in the AMY1 locus is impacting the microbiome. We see a big effect on the oral microbiome, and it looks like, and this makes sort of intuitive sense, that if you have higher amylase activity, you have breakdown of starch in the mouth, you have simple sugars that can drive these disease-associated taxa. And then in the fecal microbiome, we see Ruminococcus in its genes. We see some sort of interaction with diet for a mild mouse phenotype. And it's possible that if when people switch to agriculture, that diet change brought about a change in the microbiome that could lead them to be slightly more, have slightly more body fat. That could have been one of the many reasons why those people were selected for over time for survival, perhaps. So I'm going to stop. Yeah, I'm going to stop here. this is two different examples of how genotype, human genotype, so allelic variation or copy number variation can have some impact on the microbiome that then can feed back to affect phenotype, which is then obviously what can be selected on. And so you can imagine feedback loops here between microbiome composition phenotype and the genotypes underlying those microbiome phenotypes. So you've got a lot of G by E by P by M by, so a lot of fun things to figure out there. And so this has got us thinking, since both of these studies were looking at genotype variation in the human population that is thought to have occurred when there were very big events in our recent evolution, that a lot of which are related to diet. So fire and cooking food reduces dietary complexity. So it's a way of cooking food is a way of pre-digesting it in a sense, and switching to foods that were higher in starch, switching to animal products, switching to drinking milk that isn't from humans, switching, taking on new foods like, for instance, seaweeds. We know that some wonderful work has shown that gut bacteria picked up genes necessary to break down special polysaccharides present only in seaweeds. We know there's been epidemics, pathogens, and then more recently, we're mixing things up so much by traveling. We're basically cutting down on transmission between people with our modern hygiene. So by reducing pathogen transmission, we're also reducing commensal transmission. And so one of the things we're going to be doing in Germany is think deeply about what's happened to humans you could think of it as the recent past, so since the Pleistocene, and how selection on humans could be mediated through these interactions with the microbiome. And it's actually not an institute that I lead. It's a department, which is a fancy word for a big lab, but it's really just a lab. Anyway, so this is the kind of thing that we're going to be continuing to look into again. So these are the people that did the work: Julia Goodrich and Angela Poole and Jill Waters. So some of them stayed at Cornell. Some of them have moved over. We've got a group now, the sort of beginning of our group at the MPI, and then some of them have since moved on. And most of this work was funded through the David and Lucille Packard Foundation and the NIDDK, but it was really the David and Lucille Packard Foundation that got us started with all of this, which was really wonderful. And finally, I'd like to thank the twins who can't do any of this, and thank you for being here.
I'm wondering at the start of your talk that you have this nice variation in heritability across all of your microbes. So I'm wondering, now that there's so many non-human microbiome data sets out there, have you done a simple look to see whether the more heritable taxa seem to be more human specific? So are you less likely to pick those highly heritable taxa up in non-human microbiome data sets than the less heritable ones?
No, I haven't, but that would be interesting to look.
Yeah, it seems like it would be pretty easy with all the
Yeah.
data sets.
So, right, Kristin Slaniaceae is present at 0.1%, so the data sets have to be deeper than they used to be, so as long as they're sort of have sufficient depth. So you also got a detection issue.
Yeah.
But I do believe that many of these things are present across not just humans and mice, but you see them in other things as well. Whether they're the same ones, I doubt, but related probably.
For the Kristinaceae, are you or any of your collaborators doing any physiology experiments to test your predictions about how they might be interacting with the other gut microbes, like taking fecal sample, adding the species, and then putting it in a fermenter and seeing what you put in, see what comes out?
This is the kind of thing that we're doing now. So we're modeling the system by putting just Kristin Sinella in with one other partner that for instance, and so we're also doing not abiotics, but we're very interested querying who the partners are and seeing if we can enrich for them in bioreactor systems and so on. So yeah, we want to get at how the unit functions and what the metabolic output is, and how that affects then post-metabolism.
The labels on a lot of the slides were so small it was hard to read.
sorry.
So are there particular species level taxa that persist from the mouth all the way through the entire alimentary canal? And are those taxa performing different tasks in different parts of the digestive system?
So my understanding is that you might be able to pick them up because they're traveling through, but the ones that are at different sites are specific to those sites. And so although you might find the same genus in the mouth, or say Prevotella is a good example, you can there's a lot of Prevotella, different species and strains in the mouth, but they'll be different in the gut, and they'll be doing different things there. They have different gene content and so on. So it gets tricky when you're looking at places like the stomach, because they're the true residents, the autochthonous microbiota, so such low biomass that the tourists can look like they're really there, but the autochthonous ones look like you pick them up, but they're probably just on their way through, is what we think. Whether or not it's important for them to be on their way through and are actually doing something on their way through is another question. But it gets tricky when you want to look. The small intestine in particular is a site where we know that there's a lot of crosstalk, but knowing who's really there and what they're actually doing is difficult to access for obvious reasons.
As a follow-up, for ruminants, they often put these things in the sides of their bodies that they can go in and sample parts of the,
right, yeah.
And I don't know if that would be a useful way to determine function. Well, not in humans, of course, but unless you get a volunteer.
Yeah, there's people with ileal ports that's actually been done to. Although they have a tendency to die, so that's really it can be grim. Yeah, no, I agree. The cannulated cows are fascinating, and and there's also people developing pills that could go in samples that go down and that kind of thing. But yes, there's a lot to be done in that. Yeah.
Have you looked at people with extreme diets, like absolute vegans or total carnivores?
So I haven't, but others have, and also putting people on extreme diets for short periods of time. And you can see, so that will differentially sort of amplify different members of the community and also shift their gene expression to do different things. But diet is a huge remodeler of what's there. And probably if you want this healthy microbiome, you should eat well and exercise. That's what it's so it's not really very useful. Yeah.
So I'm thinking within the heritable OTUs, there's probably lots of strain level. Is there any interest in using metagenomics to look for like negative epistasis essentially between different strains?
Yeah, I'd love to. Yeah.
It's really expensive maybe.
Right, so this whole collection went to Craig Venture's company, HLI, but we're not going to get to see the data until they've published it, so we're just waiting. Unless we just go ahead now and redo it ourselves, which we might want to do. Yeah.
So bacteroides that you found to be not very heritable, I mean, they're responding really strong to diet. So thinking about the opposite, how do the taxa that are very heritable respond to diet? Do they respond more or less? Not at all?
We don't really...
If you look at the other data sets, perhaps.
We haven't seen an association with diet in the other data sets. But I've heard through the grapevine that Tim Spector has been feeding twins something. And but I don't know. So we don't know yet. We haven't seen it. But from what I know, they're not responding to diet. From the data sets we have access to and we can look at. So whether or not that changes with time, we'll see. It could very well, because it would seem to me that this consortium might be set up for fiber degradation, and that would make sense with leaner people on a better diet. That might all fit. that it could come down to the fact that they're just eating a more fiber-rich diet, but we don't have the data yet.
Yeah, so this is a really interesting talk. Thank you so much. I wanted to follow up on that question. When you were doing the heritability analyses, what were you actually using as your phenotype that you analyzed? Was it just a presence-absence of each of the
It's the
taxa
relative
or
abundance, relative abundance of taxa.
So out of curiosity, what's the measure? Like, what did you actually measure?
It's essentially sequence count. It's the data are sequence counts, and then the models, and so we regress out a whole bunch of things that we're afraid might influence counts. So, for instance, age, BMI is regressed out, technical things like batch effects are regressed out, and then we model the residuals.
Oh, okay.
So you started by listing these important functions
Yeah.
of the gut microbiome and said, why would an organism leave that to chance, right? And as a motivation to look at heritability. But then you found that, in fact, most taxa are not very heritable, right?
Right.
And,
The vast majority.
the vast majority.
Yeah.
And so, but presumably those microbes are performing functions that are important for the host,
Yes.
right?
Yeah. They're definitely mammal, if not human-centric microbes, for sure. It's just that the genetic differences that we see, the differentiation between the people aren't really affecting their abundances. So
Yeah.
they're there, and they're definitely not going to be. They're not going to find them out in the environment or in a fish necessarily, or they're clearly adapted to human physiology and so on, or mammalian physiology.
Yeah. Do you think is that a consequence of the sample group that you've looked at, and maybe the amount of genetic variation that exists among the dizygotic twins not being very great, for example?
Well, no, okay. I think that if you're going to do this study, this is probably one of the worst ways you could do it. So, because stool is a sort of a big mush mix of the whole community coming down the GI tract, the mucosally associated plus the food, and we know that. So we're looking at this mix, whereas you might want to be more specific with where you're looking. So that's the first thing. 16S is, as we know, a bit crude, so the strain differences and so on, that could be very important. Probably the host is more interested in function than phylogeny, and we know that function is shared a lot between microbes, and so again, if we were to redo this, taking a more functional focus, I'm sure these numbers would go up. So for me, this is a first pass, and it's encouraging. And I think that any way you focus in now is gonna reveal much more. I think it tells us that there's something there, and there's a whole lot more to find out. That's how I think of it. Yeah.
I have two questions. First, so you said you found the family called Christianaceae
Yeah.
is the most inheritable based on your data. Is that any relationship of the functions of or genes in that family which might interact with human genes, so that make it more inheritable than other microbes? The second question is that, so in the fecal transplant experiment to the mice, so you transfer the whole fecal. So have you ever isolated that any species of that family, or?
Yeah, so we do have one cultured isolate to work with, so we added in, and we're also doing work now with just it alone or it with a partner in germfree animals. So it's types of experiments we have going on now. See if we can model it, taking out some of the complexity, and why it's heritable. Well, we don't know. I mean, I think the idea that diet is somehow involved could very well be part of it, because we know that diet preference is heritable. A lot of things are heritable. So it could be that people are preferring certain foods that encourage this thing. That's possible, or it could be some other type of interaction, but what we've been able to see from the GWAS hasn't given us any clues. It's not, we're not picking up a particular immune gene or something where we might say, it's not sensitive to some antimicrobial peptide or something. We don't have anything like that to go on yet. So, so right now it's most highly heritable, but we don't know why.
Thank you.
Yeah.
Maybe one more question from Chelsea.
I was just going to ask, sort of, in a sense with that, is there anything, so is there a subset of your twins that are monozygotic but have weight differences, like significant weight differences? And would it be possible to segregate those out and look at any of these heritable bacterial part of the communities within that difference so that you could really target, you've got same genotype, different phenotype, but within just that subpopulation of the monozygotics?
Yeah, that would be interesting. I don't know if we've done that, but
do you know if there were any monozygotics with significant weight differences where that could be?
I think we've got a few, but they're tough to find. The discordant ones. I think we do. We could definitely.
I was just wondering if any of those would highlight that particular group. That might be another way to find some.
So we know that whether or not we have BMI controlled for in the analyses doesn't affect this.
Okay.
But we haven't. I don't think we've done what you're suggesting.
There is a study that was published about discordant twins, so maybe the data is out there and you can just look at it. I don't remember the authors, but there was a study.
I'm sure we have them. Yeah, well, we're going to have a subset of discordant for BMI for sure. Yeah.
Somebody's itching to ask one more question. Yeah, go ahead.
It might be hard to find discordant twins in like obesity, but you can probably find discordant twins in like one of them lived abroad for a while, or one of them like, you know, just
monozygotic twins separated at birth.
yeah, or something
Those
like
are rare.
something like that.
No, we for sure have them discordant
Environment
for
in some
BMI.
way.
So we could look. I don't know off the top of my head if they differ for Kristen's and
And sorry, would they have the same amylase copy number? Or is amylase copy number completely?
The twins?
Yeah, it's not something that's duplicated like it's
Ah,
not like a
so
somatic gene or something, is it?
right. So there's actually a SNP which is a good predictor for AMY1 copy number.
Okay.
But AMY1 copy number, you have to go in. Typically, you have to go in and measure it yourself, and there's a lot of arguments about how well people do it. So there's been some population level stuff where people are asking, does copy number give you a high BMI, for instance, over a set population? And the sets of authors are sort of butting heads over how it's done. So it's actually pretty tricky. So we've looked a little bit in the twins at that SNP that's supposed to relate, but I'm not sure if it. I'm not sure if it's really the best proxy, really, that you really have to get in there and do a really good job measuring it yourself. So for some studies that have found a link between copy number and BMI, others are saying they did it wrong.
They didn't have a proxy for copy number.
Or that they measured, when they directly measured it themselves, they didn't do it right. So it's tough. Good tough.
All right. Well, let's thank Ruth.